Hello there. This screencast is going to cover the balance of the unit from last week, cell transport mechanisms. This is where we talk about things that can go in and out of the cell itself. And then also we'll talk further on about how cells divide. So all cells are surrounded by a fluid. You remember in that video that I showed you in class, there was the outside of the cell, the extracellular fluid, and the inside of the cell, the cytoplasm. So the cells are surrounded by various nutrients like oxygen, glucose, and amino acids on the outside, and they have to be able to get into the cell inside, because they're used by the cell. Additionally, the waste that is created inside the cell must be able to get out so that they can be eliminated from the body. So there's a mechanism for transporting these products in and out of the cell that we'll look at. There are two main groups of transport mechanisms to get substances in and out of the cellular environment. When there is no energy required to move the substance, it's called passive transport. When energy in the form of ATP is required to get the substance through the barrier, it is called active transport. So think of passive transport if the substance is the ball, and just simply gravity will take it from one place to another. No energy is required. The ball will just roll down the hill. Consequently, if that ball has to get up the hill, energy is required. And for energy, we call that ATP, which is a short form for a source of energy that is created within the cell by the mitochondria, and we talked about that last week. There are different types of passive transport. One is called diffusion, where we move substances from an area of high concentration to low concentration. Facilitated diffusion, where there's a helper molecule that assists in the movement. Osmosis, which is where we can diffuse water from an area of high concentration to lower across a semi-permeable membrane. And filtration, the pushing of water and dissolved substances from an area of high pressure to low pressure area. So diffusion is the most common transport mechanism. If you look at our picture on the left, it shows the diffusion of a little red tablet, a little red dye. So the concentration of the tablet when it goes in is of course high in that location. As the tablet dissolves, the concentration of the tablet, it starts to dissolve and filter and diffuse to other areas in the beaker until equilibrium occurs. So it can't diffuse any further. So we have balance. This figure B is basically showing what would happen if a scent is passed through an area of high concentration from the skunk's tail to a lower concentration until balance is maintained. Eventually, the scent is dissipated throughout the environment. Diffusion is also the mechanism that is responsible for moving oxygen into the blood and carbon dioxide out of the blood. So those are the kinds of things that have to be passed over that cell membrane. Facilitated diffusion is when there's a helper molecule that will assist the product from getting across or moved from one place to another. and this will help to increase the rate of diffusion. Here is the passive transport mechanism called osmosis, where we move a solute in water from an area of more water to an area of less water across a semi-permeable membrane. Semi-permeable means selectively permeable. It only allows the passage of some substances while restricting the passage of others. So in this case, we've got water moving from an area of more water to an area of less water. The ability of a solution to affect the volume and pressure within the cell is called tonicity. So what we're looking at here is a red blood cell that is placed in an isotonic solution. And in the first one, isotonic, we have no net movement of the water, so it's equal. In the second picture, the hypotonic, water diffuses into the cell, making it larger and more fluid filled. And the final one is hypertonic, when the fluid seeps out of the cell until it bursts or dies. Sorry, the hypotonic one will burst and die. The hypertonic one will just shrivel up and die. So in nursing, they have intravenous solutions that are isotonic so that they won't cause damage to our red blood cells. Filtration, again, this is more for nursing, but this is the movement of water and substances from an area of high pressure to an area of low pressure. So you can see the syringe that is used, the plunger pushes the product through to the end, unless the plunger has small holes in the side of it here, and then it diffuses out through the little holes. So that's very similar to what our capillary walls look like when blood pressure occurs, the force of the blood from the heart. If there's excess water, etc., and it's happening too fast, the water seeps beside the capillaries and leaks out. So all of those were just passive mechanisms where products get from one side of the cell to another side of the cell, or within that cellular environment, from the inside to the outside, or from one part of the body to another. Now then we have active transport. Active transport, there are three main types. The active transport pump. This requires ATP to help move the substance into a cell. And then we have endocytosis, where food or substances are taken into the cell when they come along the surface and the cell membrane engulfs it, or exocytosis, where the substance that was once inside the cell in a membrane-bound vesicle, like Big Bertha that we looked at on the video, comes to the surface of the cell and then is released. The contents are released out of the cell. That's exocytosis. So the transport pumps, this is where an input of energy is required. They pump an area from lower concentration to higher concentration. This is common in potassium into a cell because we have potassium in our cells, but most of it is located inside the cell. So in order to get more into the cell, it needs help to get in there. So it requires ATP. Here is an example of endocytosis, and there are two subcategories of this. We have, if the particle itself is too large to pass through that semi-permeable membrane, the cell membrane, then it will come to the surface and the cell membrane actually will wrap itself around the product and bring it inside in a little vesicle within the cytoplasm. If it is a solid particle, we call that phagocytosis. And if it is a liquid or water droplet, it's called pinocytosis. And then the exact opposite of that is exocytosis, where we move substances out of the cell. It helps, this is what we were talking about when we saw the Big Bertha, and she's carrying proteins along the cytoskeleton and walking that sac of that vesicle of proteins until it gets to the surface, and then that sac merges with the cell wall and the contents are then released out of the cell. So those were the transport systems, and now we'll just briefly look at how cells divide. Cell division is necessary for body growth, repair, and reproduction. And there are two types of cell division. Mitosis, which is pretty much all of the cells of the body divide by mitosis, except our sex cells, and they divide by meiosis, just a different term. There are two phases of a cell cycle. The first gap phase, the cell carries on basically normal function and begins to make substances inside that gets the cell ready for cell division. It will duplicate its chromosomes and prepare this and synthesize enzymes and other proteins that are needed. And then it will move into the final stage of mitosis. In mitosis, there are 4 phases: prophase, metaphase, anaphase, and telophase. I apologize, my diagram here, I just have to move that out of the way here for a moment so we can see the words. There we go. And there's a great analogy playing. One moment. Okay, sorry. There's a great analogy that helps you remember it, and if you think of play me a tune. This play refers to the prophase, and in this phase the chromosomes coil tightly. Me, or metaphase, the chromatids align in a narrow central zone. Anaphase, the centromere splits and chromatids pull to the opposite poles. And tune, telophase, new cells revert back to the interphase stage, and then a brand new nuclear membrane reforms. Chromosomes uncoil, chromatin strands reappear. So basically the cell cycle has 2 phases: the interphase, where the cell continues to function but prepares for division through growth and DNA reproduction, and mitosis, that play me a tune, that actually one cell will divide into 2 daughter cells that are identical genetically, and it goes through the prophase, metaphase, anaphase, and telophase. All right, so that's all there is to review for that portion, and feel free to play this as many times as you would like, and I'll see you on Friday.